Hi guys, and welcome to lab this week. We have 2 lab worksheets to get through this week. I realized that in your lab schedule, some of the weeks that we went through, some of the labs kind of doubled up, and I wasn't really doing that. So that's why maybe some weeks seemed really light for lab. So we kind of have a little catch-up work to do, but not really much. Just this week and next week, you'll have double labs. And it still will be really simple and really easy to do, but I just want you guys to note that you'll have 2 worksheets to complete this week. 2 worksheets to upload. It'll be on lab 14, which is the digestive system and nutrition, and lab 15, which will be reproductive and urinary system. And I'm going to briefly go through both here, and they're pretty self-explanatory. There's a lab guide that goes through both of them. In lab 14, the digestive system, for example, the first couple parts will be labeling the digestive system using the following terms, and then filling out this chart, which will describe the structure as primary or accessory. It's primary if it's a part of the digestive tube, and it's accessory if it secretes digestive fluids into that GI tract. And then you'll want to write the location and the function. So that's the bulk part of this lab. Emulsification, you'll kind of read through, you'll watch a video or link. Emulsifying has to do with how we digest fats in our body using bile. So that'll go through what emulsification is. And then you have a couple questions to answer based on that. And then you have a little mini experiment to do in your home, which you should be able to do, showing the effects of enzymes. And enzymes are really important in the digestive systems because they help catalyze reactions. And they're especially important in the digestive system because they help these reactions of digestion go much faster. Specifically, enzymes lower the activation energy. So what that means is they just make it easier for a chemical reaction to occur. And this shows a little diagram of the enzyme and the substrate, the enzyme substrate complex. And then the enzyme, which as that bottom part, will always be unchanged at the end, but 2 new products will form. So that's kind of how the enzyme works, how it binds with a substrate or a molecule, and then it'll form new products at the end, but the enzyme will go unformed. So you will be doing a little experiment with the apple and seeing how changes in environments, such as temperature, pH, lack of oxygen, will alter the color or browning of the apple. So you need 1 apple. You can also use a potato. You need 4 small dishes for holding individual slices, a grater, lemon juice, water, ice in a larger dish with a smaller dish set inside of it. And this will be your cold intubator, a warm water bath with a smaller dish inside of it. And this will be your warm water intubator or incubator. And then you'll assemble the following experiment using, before you cut up the apple, you'll have a cold intubator as described above, the warm intubator, a dish with lemon juice with water, a dish for the grated apple, and a dish for an untreated apple, the control. So you'll get 6 slices of your apple or potato and put 1 slice in each of these 6 kind of containers. You'll check the apple for every 5 minutes for a total of 30 minutes to determine the intensity of the browning. So you'll complete this kind of chart. 0 means no change in color, and 10 means extreme change in color. And you're just going to rate your fruit section based on the dish it's in, whether it's warm, cold, lemon, grated, nothing, water, after 5, 10, 15, 20, 25, and 30. So you're just going to rate kind of the browning or change in color of that apple slice. Then you'll kind of, for each of the treatments used, indicate what condition was caused by the treatment and how the condition affected the reaction rate. So for example, what did ice do? Well, that would be a decrease in temperature. And did that increase or decrease the enzyme activity? Meaning, did that increase or decrease how fast or quickly the apple browned? And you'll go through with that chart. You'll identify 2 sources of amylase in the body. So you might have to do a little research there. What does it mean when an enzyme is denatured, and why is that bad? What can denature enzymes? And in which treatments was the enzyme denatured, and how do you know? And explain why it's important to maintain homeostasis for the function of enzymes. So for this, you also need to show me a picture of your experiment. So when you're done, before you clean up, just take a picture of your experimental kind of setup and place that in the lab document as well. And that's lab 14. And then lab 15, and I'm not really going through the lab guides, but the lab guides will help you give a little more kind of instruction as you do that. Lab 15 is shown here then. And lab 15 is all about your urinary and reproductive systems, and it's mainly just labeling structures in these systems. So you can use your lecture notes, your textbook, to label the structures of the excretory or urinary system, labeling the specific structures of a kidney, talking a little bit about buffers, and this will take you through an experiment, but we provide a lot of the data for the experiment in the lab guide. So be sure to look at the lab guide for this part of the experiment, but it will be talking a little bit about buffers. Specifically, we give you the pH of this chart to fill in, and how does an effective buffer respond to added acid or base substance. What a buffer is it resists changes in pH. So you should see here that the buffer, the starting pH, the pH after adding an acid, and the pH after adding a base should be relatively constant, because that's the point of a buffer. And buffers are really important in our body because they can resist changes in pH. Then we'll do a similar procedure. Again, this procedure is outlined in your lab guide to test for 3 substances found in plasma, protein, bicarbonate, and glucose, to determine their effectiveness as a buffer. So you'll look at the starting pH, the pH after adding an acid, and the pH after adding a base. And the best buffer will be the one where the pH stays relatively constant, because that's what buffers are important for. They resist changes in pH. So you have a couple questions to answer there, and then you have some labeling to do of the reproductive system, the male reproductive system, then the female reproductive system. And then you'll go through and list the structure and function for each of the male and female reproductive systems. There's a couple questions to answer at the end, and then that takes us through lab 15. So again, just a reminder that you have 2 lab worksheets to complete and submit this week. And your quiz will be on both labs for next week as well. So take care, guys, and thank you for listening.